vaccination in sheep and goat. If we talk about the scientific sheep and goat rearing, it will depend upon four important pillars are there. Number first is breeding. So breeding, all of us know that if we have got a good breed, half the job is done. Now, the second thing which is important for rearing of a sheep is feeding. Depending upon the nutritional requirements of a particular animal, that may vary. Third is the management. How better we manage a particular farm, be that housing, be that feeding, and be that any other management which is related to the farm. So that has to be taken care of. That is the third aspect. Health is the fourth and most important aspect of a pillar of a proper, if we are talking about rearing scientific sheep and goat rearing. Once we talk about the health, we know that animals, they suffer from various diseases. They suffer from infectious diseases, then they suffer from the non-infectious diseases, and both of them, they result in loss of productivity, and which in turn they result in less economical farming practices. So it is better, if we are talking about the health of a particular farm or health of these animals, it is better that prevention is better than cure. Rather than curing an animal for a particular disease and spending extra money on treatment cost and other things which are there, we must focus on how we can protect an animal from from diseases who are at a risk of particular disease. How we can, when I'm talking about the disease here, I'm talking about infectious diseases. Since we, in case of infectious diseases, we talk of a vaccination program. So it is by means of vaccination, by means of vaccines, we can protect the health of animals, that sheep and goat, and that can result in a successful sheep farming or successful enterprise. So vaccination is one of the most important aspects of maintaining a particular good health in a farm. So that has to be taken care of. So now we maintain a good health as prophylaxis to protect them from particular diseases. We maintain them by the use of vaccines. Before going into the vaccination and before going into the different aspects of vaccination, we must know a brief about history, where from it has come. Basically, we are here in the modern era where we have vaccines of different types available with us, and we have an example of COVID-19 vaccine which was developed very swiftly. We were not even we never imagined that that vaccine will be prepared so swiftly, but across the globe, scientists and researchers, they prepared vaccines for COVID-19, and even in our India we had our own indigenous vaccines. You know, when we talk for vocal for local, so we did that and we prepared it in our own country. So that's how the science of vaccinology and vaccine, it has developed over the period of time. But credit must go to those who basically started it from scratch. They Nobody knew about the vaccines when these stalwarts, these scientists, they just applied their mind and they applied their genius intelligentsia, and they came up with these fascinating inventions and discoveries and other things. And credit must go to the one, we everybody of us, we must be knowing about the name Edward Jenner, who was a English physician, who actually the word vaccine, he gave, it came from a lated variola vaccinia, whereby he used this cowpox to treat the humans for smallpox disease. At that point of time, you imagine in 1798, what would have been the era of science at that particular time. But he applied his mind. Basically, before that, they were using this variolation. They used Lady Mortigo. She used that dried scab culture from that human smallpox, and they used to inoculate in the humans, and that was the before this use of cowpox against the smallpox came into existence. Then Edward Jenner, he basically started it, and he used cowpox for the as against the smallpox. That was the first of its kind. Then with the advent of vaccine now, we know what actually vaccine is, and it is basically whatever the biological preparations we prepare out of the living organisms. By living organisms, we mean it may be a bacteria, it may be a virus, uh, by whatever the biological preparations we prepare, and they enhance the immunity against diseases, thereby preventing a particular disease, which we call as prophylaxis, prophylactic vaccines, or in some cases that can treat the disease as well, therapeutic effect can be there. So that is what we call it as vaccine at this now. So vaccines, we know they can be administered in many forms. They are available. They are available in liquid forms. They can be injected by we can have injectables, oral or intranasal vaccines. So this is on your screen. This is Edward Jenner. During our programs, this graduation programs, we have read about this Edward Jenner. He vaccinated this James Phipp. He was a boy. He vaccinated him against this. He used the cowpox to vaccinate against the smallpox, and he gave him a challenge with the infectious agent, and the boy survived. That was an experiment which changed our world. And thereby before that, variolation, as I told you, that variolation was practiced in ancient China. It was popularized by Lady Mary Montego. Then after Edward Jenner, Louis Pasteur came. He was a French chemist and microbiologist who made some of the key discoveries, and he worked extensively on vaccine development. He discovered the germ theory of diseases and principles of vaccination. Then he was the person, Louis Pasteur was the person who made the first vaccine against some of the very important diseases in humans as well as in animals, that's rabies and anthrax. Louis Pasteur was the man who made these vaccines. And then because of this vaccination which came into existence, we were able to eradicate the smallpox in humans in 1979, and in animals through extensive vaccination and other practices we were able to eradicate rinderpest in animals in 2011. We can never imagine eradicating a particular disease without involving vaccination in it. So vaccination has the potential to eradicate a disease completely in particular region. So that is how the vaccination, how important the vaccination is. So these are a few of the scientists who changed our future. There are different types of vaccines. Briefly, if I can give you an overview, that there are different types of vaccines which are available. One is that we have live attenuated. What do we call it as live attenuated? Basically, what vaccines are? Vaccines are the nothing but these are the organisms which are weakened over the period of time. They are weakened. Their virulence is decreased. Their pathogenicity is decreased. They are able to, these preparations are able to elicit an immune response in an animal without causing a disease in that particular animal. The pathogenicity is not there, but the immune response, they are able to generate the immune response once we inject them into an animal or into a human. So there are different means, and there are different means and ways how we can prepare these vaccines, how these vaccines are being prepared in the laboratory. Number first is live attenuated vaccine. As the name suggests, indicates that these are live vaccines, but these are attenuated. Attenuated means their virulence is decreased. How we decrease the virulence of these vaccines? There are some ways and means, laboratory mechanisms by which we can decrease the virulence of these. For example, we give them some suboptimal conditions. I will quote an example here. If we talk about the Mycobacterium tuberculosis vaccine, BCG vaccine, how that has been made. The BCG vaccine, basically the Mycobacterium tuberculosis, that organism is grown in a bile-rich medium over a period of the bile-rich medium. Basically, it's not a favorable medium for that, but it is some suboptimal conditions are given to that. It is grown in that bile-rich media over a period of time, over 13 years period of time. Scientists, they could see that while growing in that bile-rich media, it has lost its virulence. It's no longer able to cause disease in a particular individual, but once we inject it in a particular individual, it's able to cause produce the immune response. So it is, we can use as a vaccine, we can use this candidate as a vaccine, we call it as a live attenuated vaccine. Similarly, we produce different types of attenuations are being carried out by growing a particular organism at a different temperature than it is optimal temperature, or we can produce a particular by serial passaging in cell culture. We can produce this live attenuated vaccines by different serial passaging in cell cultures, by which the particular vaccine, particular virus loses its virulence, and then but it is antigenicity is maintained, it is able to elicit an immune response, and then it is used as a vaccine. So that is live about the live attenuated vaccine. These live attenuated vaccines, they have an advantage that they produce a good amount of immune response. Once we inject them, once we use them as a vaccine, they produce good amount of immune response in an animal or maybe in a human. But the problem in that is that they can revert at some point of time. It has been seen the a disadvantage associated with them is that they can revert, means they can revert back from virulent, they can get back to the virulent stage. Particularly, we avoid these type of vaccines in pregnant animals because of this disadvantage, that they can revert back to the virulence and they can actually cause the disease. But those uh, reversion processes are very rare. But that can be a possibility. So that's a sort of disadvantage. But those possibilities are very rare. Otherwise, they produce a good amount of immune response. They produce good. We don't need booster doses in case of live vaccines. That is an advantage because they produce a good immune response, they produce good memory cells, and in that case, we don't need booster doses in case of live vaccines. Number second type of a vaccine is an inactivated vaccine. Inactivated in other we use synonym for this is the killed vaccine. In what we do in this is that an organism is killed, a bacteria or a virus is killed, we kill that organism is no longer able to replicate inside a body, but it is antigenic structures which are on the surface antigens, they are maintained, the structure of those antigens is maintained, and once it enters into the body recognizes this antigen as foreign, at it produces an immune response against this particular antigen. And but this killed this vaccine, they, whether it's a bacteria or it's a virus, it will not be able to replicate since it's a kill. So kill type of an organism then. So again, the process by which it's been made, there are several the chemical treatments, like we use formaldehyde or we use beta propyl and ethylenamine. These are the chemicals which you use for killing, or we thermal killing can also be there. So it varies. So we kill the organism, but its antigenic characters, antigenic structures which are on the surface, they are maintained. There's no change in its antigenicity. And once we purify it, then we can use it as a vaccine. As I was talking about at the start, we were discussing about the foot rot vaccine. I was discussing with Dr. Ashok that we at Division of Veterinary Microbiology and Immunology in our own division where I'm working, we have developed a vaccine against foot rot, which is a killed vaccine. We use formaldehyde for killing of that. This we have selected a particular strain. We grow them in a broth culture under anaerobic conditions. Then we kill the organism. And but here. The issue with inactivated vaccines or killed vaccines is that they do not produce that sort of immune response. The immune response is not as high as in comparison to that of live attenuated vaccines. That's why we need to give booster doses. Once we use, mostly when we use kill vaccines, the booster doses are to be given there. But what do we mean by booster dose? I'll come to that later on. But we need to give booster doses so as to increase the increase the response, increase the antibody titer in an animal, so that we need to give in case of these inactivated vaccines. It's a must that we need to give the booster doses. But the advantage here with this is that they will not revert back. They will not. There will be no virulence associated with them because they are killed, and there is no possibility of reversion back to virulence. So that's an advantage with these activated vaccines. But usually, in case of these inactivated vaccines or the kill vaccines, it's so as to increase the this immune response, we use adjuvants. We use different kinds of adjuvants to increase the immune response. But that's not the case of live attenuated vaccines. Why we don't use any kind of, normally we don't use this adjuvants with that. But here we need adjuvants to increase the to improve the immune response once we inject them into an animal or into a human. So these are the main, these are the two main types of vaccines, kill and these attenuated vaccines. Now we have another vaccines which we call as the recombinant vaccines, recombinant subunit vaccines. Once I talked about the attenuated vaccines or the killed vaccines, in both of these types, we use the whole organism. We use whole bacteria or whole virus as vaccine. But once we talk about the recombinant vaccine, we don't use whole organism as a vaccine. What we first do is that we try to find out that in a particular organism or in a particular bacteria or a virus, what is the immunodominant protein? Immunodominant protein means the protein, surface protein or antigen which against which the antibodies are produced inside the animal, in the body of an animal or in the body of a human. We identify that immunogenic protein. Then we identified the gene associated with that immunogenic protein, because a protein is being produced ultimately, it's a gene which produces a protein. We all know about that. So we go to the gene level. We find out, we have different tools are there to find out this thing, what's the protein involved in this, which is immunogenic. Then we find out the gene which is involved for this. We isolate that gene, we excise that gene, clone that gene, and we express that gene in a particular cell, that we express that gene in a particular cell. Then that after the expression there, the protein is being, we produce the protein at a very large quantity of protein is being produced. Then we purify the protein and we use that protein. For example, if I talk of subunit vaccine, we can use that a protein we can use as, once we purify that protein, we produce, the mass production of that protein is done. We produce that and we use that and we purify that, and we can use that as a vaccine by you, with the help of adjuvants. We can add different types of adjuvants like aluminum adjuvants or aluminum hydroxide, we can, and other types of adjuvants are also available. So we can use that in that subunit vaccines. Other type of recombinant DNA technology is that we isolate a particular gene. We know that this gene is responsible for this is the immunodominant gene. This is the gene which is antigenic in nature, which produces a particular protein which is involved in this immune response. Then we isolate a particular gene. We use a different kind of a vector. We call that as a vector. For example, if you talk about the COVID-19. In COVID-19, we first we try to know that which is the most immunodominant protein. And once we discovered that immunodominant protein, we isolated that particular gene, and we use that in adenovirus. That is we call that as a replication deficient adenovirus. We introduced that gene in that adenovirus, and that adenovirus we used as a vector. Once we introduced that gene in that adenovirus genome, that adenovirus expressed that particular gene along with its own genes. That's a replication deficient adenovirus. Once we introduce a foreign gene into it, it becomes replicative competent. Then once we use that as a vaccine, then it no longer produces a disease, but it is able to produce those proteins which against which the antibodies are produced. So we can use that as a vaccine. So there are different vaccine recombinant DNA technologies which we can use for vaccine production. This is the modern methods of production of vaccines. Again, there are some issues maybe at some point of time. The issues associated with subunit vaccines is that they do not produce as good as immune response as it being produced by the live attenuated vaccines, because here we use a particular type of a protein. But if we talk about live attenuated vaccines, there are n number of proteins. It's not only a particular protein against which immune response is produced in the body. For if a bacteria is there, there may be multiple epitopes are there, multiple antigenic sites are there. So against the multiple, once there is a whole organism is there, the antibodies will be produced against the multiple these antigens. So there will be a good immune response. But if we are using a single protein subunit vaccine, when we talk about the subunit vaccine, if we are using a, for example, we have a FIMRI, here we are working on a subunit vaccine in our own lab. We are working on a fimbrial protein. In case of the foot rot, there is a protein which we call as the fimbrial protein, FIM protein, we call that as. We know that the immune response is being produced in an animal against that FIM protein. So we are trying to isolate that protein. We are trying to purify that protein. Once we purify it, we will use the adjuvants and we will go for the trials. But that depends. Maybe we will not be able to get a proper immune response because we are using is only a single kind of a protein there. So that is to be seen. But that can be a problem with subunit vaccines, that optimum level of immune response is not produced. But in some cases, we are able to produce an optimum immune response. It again can vary with the technology we use or with the type of bacteria or the virus we are using, or there are so many factors into it. But this is basically the technology which is with us. Then the fourth is the toxide vaccines. Toxide vaccines is that once we, for example, a clostridium is an anaerobic bacteria, it produces toxins. Those toxins are lethal. They cause pathogenesis. But if we use certain chemicals, we can reduce the toxogen. We can reduce their pathogenicity, and we call it as toxides. They are immunogenic, but they are not pathogenic. Then they can be used as vaccines. We use in association with clostridium, for example, when we use this vaccine against enterotoxemia, in that we also use the vaccine with this toxide vaccines in combination with that. Yeah, clostridium tetani is there. It produces, particularly it's in case of clostridium tetani, we use the toxides. So this is again another type of this vaccine which we use. These are broadly four types of vaccines. There are other vaccines, genetically engineered vaccines, modified vaccines, which are there, but I don't think that's needed here. That's not needed because actually we are not discussing about. We are discussing vaccination sheep goats. So whatever the vaccines which are commonly used in sheep goats in this era, we are going to discuss about that. But basically, we must have a background about how they are being produced and what is the advantage of one vaccine over the over vaccine. That's why I discussed briefly about these types of vaccines. So now the second thing is that a vaccine, it can be a protein. Once we talk of antigen, all of us, we have read about the properties of an antigen. How a good antigen behaves. What are the properties of a good antigen? So a good antigen is, if we talk about a protein, if protein is a good antigen normally, if we use a protein vaccine, there will be a humoral immune response inside the body. Humoral immune response means antibody mediated. There are two types of immune responses. Broadly, there are two types of immune responses within our body. Once an infection, natural infection is there, or a vaccine is introduced into the body, injected in the body, we can expect two type of responses. One is humoral immune response, which is an antibody, where they are the production of antibodies and memory cells and all that we call it the humoral immune response. The other kind of immune response is cell-mediated immune response, where the cells are involved. Like there are T cells involved, T cytotoxic cells, T helper cells are involved. Usually, the cell-mediated immune responses are associated with viruses. Once the viruses enter into the cell, they are intracellular pathogens. They are found inside the cells. Once they enter into the infect the particular animal, will expect that there will be a cell-mediated immune response against that virus because it's an intracellular organism. Normally, in case if a bacteria, usually a bacteria is an extracellular organism, except few bacteria like Mycobacterium or Brucella, where they are intracellular organisms. In case of these normal bacteria, we expect humoral kind of an immune response. If a protein is, we use a protein as a vaccine, so we'll expect a good amount of humoral response, and there will be memory cells also in the infants. Likewise, that will be in the adults. On the other hand, the polysaccharides, they don't act as good vaccines. They don't act as good antigens. there is a humoral response is there, but there is no memory cells are not produced when we use polysaccharide as vaccines. But if we use these conjugate of protein and polysaccharide, the third one is conjugate of protein and polysaccharide. That serves as a better vaccine, where there is a humoral response as well as there is an this immune memory is also there in infants. So what happens when there are two aspects? One is natural infection, and other is vaccination. If a disease causing organism infects our body or infects an animal, that organism can be a bacteria or it can be a virus. We call that as a natural infection. Once it is there, there'll be a disease. There'll be, by disease, we mean that there will be an immune response against that in particular bacteria or against that virus. And that immune response, if that is an immune response is there, the bacteria will try to uh, you know, you know, cause it is pathogenic lesions, cause its own pathogens, and our body will try to produce an immune response. Since it's a foreign body, the foreign bacteria or foreign virus has entered into our body, our immune system will try to defend our own body. It will try to just kill that organism. Uh, but what will happen, the outcome, there are multiple outcomes. If our immune response is strong, if we are healthy and our immune response is strong, we will be having a recovery within a short period of time. Our immune system will be activated within a short interval, and it will produce its by its own mechanism. It will be able to kill that organism, and the organism, it will not cause a major harm in the body. That is in case immune response is proper in the body. The second thing is that if in many cases, if the agent is very infectious, if the organism is highly infectious or lethal, or if a person is immunocompromised, in that case, or if an animal is immunocompromised, in that case also we can expect that it can also result in death of an animal in that case. And the third thing, what we can expect out of is this natural infection, there is disability, and this organism, it can cause temporary damage to permanent damage to certain organs there, and that can result in disability. That's the third. And apart from these, it results in immune memory. That means our body, it recognizes, it produces those type of cells which can recognize this foreign body once it enters for the second time in our in our body. If there's a primary immune response, there is a secondary immune response. I think most of us will might be knowing about what we mean by primary immune response and what we mean by secondary immune response. Secondary immune response is that is mediated by these memory cells. Memories, I'll discuss that. I'll try to discuss that later on. But this is what we expect when there's a natural infection. When there is an infection, when there is a disease which is being caused by a natural bacteria or a virus. But when we use a vaccine antigen, or when we go for the vaccination, then obviously there will be no disease under normal circumstances. There will be only, because the pathogenicity has been decreased, the organism is no longer pathogenic, either it is attenuated or it's killed or it's a subunit vaccine, then there will be immune response will be produced within that animal, and that will give immunity against the disease. If the animal is at risk of infection, if it has got the vaccine, it will no longer the organism will no longer will be able to replicate inside the body of the animal, and it will be protected against against a particular disease, and it has a memory cell. Immune memory is also established in that animal. So again, this is a brief about how antigens, once an antigen enters into the cell, it has different, it may be a bacteria or it may be a virus. Once it enters into our body, a disease causing agent, it has got antigenic sites, what we call as the epitopes on its surface, against which the immune response, the immune system of the animal, it will recognize that whatever the antigen is entering into the body, it is the foreign antigen. It's not its own structure, body structure. It's something which has come from outside, and its immune system will get activated. There are the once we talk of humoral immune response will be developed there. There will be B lymphocytes, B cells which will be activated, and they produce antibodies, and these antibodies which are specific to that antigen, they will with the help of other cells, they will form a complex around that, and there are different complement system which will be activated, and with the help of macrophages and other cells, that antigen will be destroyed by phagocytosis, and there's a proper mechanism involved in that. I'll not be able to discuss in detail how immune system works, how the phagocytosis is completed with it, but this is briefly to show how it works, that it destroys that organism which is foreign by phagocytosis. Or in other cases also, there is antigenic recognition, there is immune system activation, antibody. This is again by phagocytosis. This is I'm talking about the humoral immune response. I have not discussed here about the cell mediated immune response, where there is the antigen enters into the body, that antigen undergoes fragmentation, then it enters into we call it the antigen presenting cells. Then antigen presenting cells, they express these antigens along with major histocompatibility complex. We call that as MSC complexes, MSC1 and MSC2. Then there are T cells, T lymphocytes, T cytotoxic T helper lymphocytes involved in that cell-mediated immune response. Against the viruses, there is a separate one. Against the extracellular bacteria, the separate one is there. So that's against. I'll not be discussing in detail about that cell-mediated immune response. That's again another kind of immune response. But here we are talking about the cell-mediated. So normally there are two types of immune responses. Once an agent enters, one is active immunity. Second is passive immunity. By active immunity, we mean once there is a natural infection, infectious agent enters into the body of an animal, the antibodies will be produced. There will be an immune response will be produced. That's called as the active immune response. Or we can produce this active immune response by use of vaccines like attenuated or killed vaccines or recombinant vaccines. Whatever immune response there is produced, we call that the active immune response. Number second is passive immune response. In passive immune response, the antibodies are not produced directly in the body by the infectious against the infectious agent. For example, if we talk about the best example of this passive immunity is this maternal antibodies. The maternal antibodies, they are transferred from mother to the offspring. During at the time of in the initial phase of the birth, if the mother is vaccinated, the antibodies which are in its they get transferred into the child also. So that's the maternal antibodies are being transferred to the child, and that protects the child for certain period of time till the immune system is properly developed in that particular newborn. So that antibodies are not directly produced in that newborn. So that's a natural infection. Or in case of snake venom, we use this. That's also an example of passive, when we use that. That's this passive immunity. This is an example of herd immunity. Most of us, we have heard about this herd immunity. This is the slide which will give you a clear idea what we mean by herd immunity. During the COVID times, we all must have heard about this herd immunity. Everybody talking about the herd immunity. What do we mean by herd immunity? Is that there are different pictures which are shown on your screen. If we talk about the first screen, the person vaccinated. In this, you treat all these dots as the animals. This is a flock of animals. If none of the animal is vaccinated there, we treat it as that person vaccinated. None of the animals is vaccinated. So the chances of an infectious agent of getting transmitted to other animals, see how fast the disease can spread. This is the red indicates that their transmission chances are more, because every animal is at risk and every animal can serve as a source of infection. That's the in case of first first case. In the second case, only 25% of the population is vaccinated. Here you can see some yellow dots which are immune. those have been vaccinated, but still there are some population which serve as the source of infection, and again there are certain population which has not been vaccinated and which can get the infection from those which are at source. So again, there are high chances of getting the disease getting transmitted over a short period of time. Then there's a third percentage, which is a 50%. These animals are vaccinated there. Again, the yellow dots are more there in comparison to the first and the second. That means the more number of animals are protected as compared to the first and second, and the transmission chances are have decreased. And then we go to the 75%. You can see the yellow, the immune animals, the number is increasing and the chances of transmission are decreasing. So with increase in percentage of vaccination, the chances are decreasing. And when we vaccinate 90% of the population, there are little chances that this disease or the infectious agent can transmit the disease and it can cause disease in a particular herd. And if 95% of these animals are vaccinated, then there are very little chances that this disease can spread in a particular flock. So that's what we mean by herd vaccination, that some population, a certain amount of the population needs to be vaccinated. It's not that we only vaccinate some of the individuals in a particular flock and leave the rest as such, but we need to vaccinate them so as to develop a herd immunity and so as to decrease the chance of an outbreak or decrease the chance of transmission of an infection within that flock. So that is to show you that how this herd immunity works. Again, this is the I've discussed this about first exposure to disease. Here what I want to just show you is that when there's a first exposure, the first exposure to a disease causing organism is there, that it enters into the body for the first time, it will produce the immune response, and that's called as the primary immune response. And there will be, if it apart from the primary immune response, it will also produce the memory cells, those cells which will recognize this particular organism. They will not produce the antibodies, but they will memorize that this is the structure of the particular organism which has entered into the body. If the organism enters next time enters into the body, the same memory cell will recognize it very swiftly, and it will produce a heightened amount of immune response against that, and it will immediately kill that organism because the memory cells are always there, already there. In case of the primary immune response, it takes a bit of time to develop an immune response. Once an infectious agent enters into the body, then the immune system gets activated. But to get the immune system activated and to produce a particular immune response, it needs, the body needs certain days. It will not produce it immediately within seconds or within minutes or within hours, but it needs, after the infection, the body needs certain amount of time, certain days. It needs to, so that the proper immune response is produced. But once a proper immune response and a memory cells are produced, once there is the second infection, the memory cells are always there. They immediately produce the antibodies against that particular organism which has entered for the second time. So that's the activation of immune. When the organism enters for the second time, there's the activation of the immune memory cells, and there will be rapid recovery. There will be no disease in that case. In the primary, there can be recovery, there can be death, and there can be disability also. So this is an important figure which all of us must know. Primary, those working in the field as a field veterinarians, they must be knowing it. But this is just to refresh their knowledge, because over the period of time when we are involved in at the field level, we try to forget these small things. But I think these kind of webinars, these give us an opportunity to refresh your knowledge. Otherwise, all the field veterinarians we have read during our graduation also, these things we have already read, but somehow we need to refresh these things. So it's a refreshing, a sort of refresher webinar, rather than a new thing for you, all of you. So what I want to show in this particular slide is that we have on the one side we have antibody level, arbitrary units, log scale. Then on the other we have number of days. We start with 0 days. For example, if we vaccinate a particular animal at 0 day, we consider 0 as a day. that we vaccinate an animal against a particular type of a disease at 0 day. So it's not that immediately there will be production of antibodies. The body will take certain amount of time to get activated. The immune system will get activated. There will be a proper mechanism by which the immune system will be activated, and there will be cells, immune cells, there's B cells, T cells, and other cells which will be activated. So normally within 4 to 5 days, for around 5 6 days, we expect that the antibody will titer will start rising, and the antibody titer will start rising, and it will, over a period of around say 12 days, 10 to 12 days, there will be maximum primary immune response. That there will be immune response will be highest at 10 to 12 days. Again, it will start decreasing. Then this immune response will decrease over the period of time, particularly in case of when we talk of killed vaccines. It will decrease, it will follow this trajectory, and it will decrease, then it will remain at a constant level over a period of time from around say 17 18 days till, and that is the time during which we need to give booster doses. When at certain point of time, normally we give booster dose at 3 to 4 weeks of age. When we give the booster dose, the memory cells are activated. Once the memory cells are activated, there is a heightened immune response. You can see the second curve. You can see a sharp curve there. That is the secondary immune response. This immune response is because of the booster doses. The first dose which we give at 0 day, it's known as the primary vaccination, is the first. And the second vaccine, particularly in case of the killed vaccines, which I discussed initially, I discussed why we need these booster doses, particularly in case of the killed vaccines. This is because there is a decreased antibody titer over a period of time. Then we need to give booster doses so as to increase, so as to activate the memory cells. Once the memory cells are activated by booster doses, which are given at 3 to 4 days of age in case of killed vaccines, then there's an increased level of immune response. There is a sharp increase in the antibody titer. Then it's a good amount of antibody titer, and this titer is then protective. That means at this level, if the antibody level is this much high, the animal will be protected against a particular disease and against that natural infection only when the antibody titer is high. So we must give booster doses. Normally we avoid in case of uh, field conditions. There are so many issues. I have been in the field as well. I have served in as veterinary assistant surgeon as well. Because of the shortage of the vaccines, we usually we at certain point of time in certain areas. I'm not talking about that it's a routine everywhere, but in certain cases we uh, feel that there is a shortage of a vaccine, and we usually do not go for the booster doses. But from this particular slide, you must understand that what how important is the booster dose. If we are giving the in case of kill vaccine where the booster dose is recommended, if we are not following the booster dose, then there is no there's no use of the primary vaccination. It makes no sense. It will not protect that animal from a particular disease. The booster dose is that important. We must follow the recommended schedule of a particular vaccination, so as to so as to achieve our intended goal. The goal is to protect the animal against that particular disease. So that will be only achievable if we use the booster doses. So we must not miss the booster doses, and we must remember that booster doses from this figure, it must be clear to you that the booster doses, why we recommend the booster dose at around 3 to 4 weeks of age interval, that we 21 days onwards we recommend the booster doses. This is because of this reason that at that point of time, the antibody levels have come down to a constant titer, which are not protective, and we need to activate the memory cells by the use of booster dose. So that's why we recommend the booster doses in that case. So again, there is a natural infection which will produce the immunity. There's a vaccination, it will produce the immunity, but the vaccination, it will not endure the illness. It will not call the illness. There are low risk of adverse reaction, but there are sometimes there can be an adverse reaction in case of vaccines, but those occasions are very rare. Even if there are adverse reactions, they will be very low. They will be of mild nature in under normal circumstances, and they greatly outweigh the risk of complications caused by the natural infection. When we compare a natural infection and with that of the adverse reaction, that it overweighs the low risk of adverse reaction overweighs the risk of complication which is caused by the natural infection. So now briefly, there are, with the example, I will try to just show you that what are the types of vaccines different. I talked about the live, killed, subunit, and toxide vaccines, but here I have given an example that what's the type of vaccine which we use in sheep and goat, which type of vaccine. For example, PPR and Capipox vaccine. These are usually live attenuated vaccines. I told you that live attenuated vaccines have got the advantage that they produce broad immune response, humoral as well as cell-mediated immune response. Low doses are needed to elicit immunity because they are highly immunogenic. So low doses are needed to elicit that immune response, and they have long-lasting immunity as well. And disadvantage is that what we can expect is that there can be adverse reaction. It can revert back to virulence. There's safety issues involved with this live attenuated vaccine. They can revert back to virulent form or wild form. I have discussed about that disadvantage as well. And in case of kill vaccine, we have an example. FMD is an inactivated vaccine which we use at is a polyvalent vaccine. It has got the advantage that it is same in pregnant animals. Usually in pregnant animals we recommend, if it's a kill vaccine, then there's no issue in that. It can be recommended in pregnant animals as well, but it has got the disadvantage that multiple doses are needed, low immune responses there against the killed vaccine, and they are highly antigenic. We need it antigenic concentration. We need in higher concentration. We need more amount of antigen to have a proper immune response. So these are some of the disadvantages which are associated with killed vaccines. Again, the subunit vaccine is that we have against FMD. We have VP1 protein. Subunit vaccine is also available. It's literally easy to produce, better tolerated, but it has got a disadvantage that induces little cell-mediated immunity, which I was discussing about you, that it is the whole cell vaccines have got the advantage that they produce a better immune response as compared to this. Every type of a vaccine has got its own advantage as well as disadvantage. Then the third is the toxoid vaccine, tetanus, Clostridium tetani vaccine. It is safe and no possibility of reversion is there, stable and long lasting, but it needs adjuvants. So as to increase its immunogenicity, it needs adjuvants and needs several booster doses also. So currently we have the vaccines commonly available. There may be, it varies from region to region. It depends upon the type of vaccines which we are using here in Kashmir, may be different from those what we are using in south part of our country. Because it all depends upon the prevalence of a particular type of a disease. We usually use a particular vaccine only when a disease is above a certain prevalence mark. Otherwise, there is no need to use a vaccine. Only when we find that the disease is crossing a particular prevalence rate, and it's becoming economically disease is very economic burden, it's then only we use that. So normally in sheep we use the vaccines in here against some important disease like PPR, sheeppox, goatpox, blue tongue, FMD, and rabies. And against the bacterial diseases, common vaccines which we use here is and against the enterotoxemia, ET vaccine, tetanus, brucellosis, and anthrax. Anthrax we don't use here. It's recommended in endemic areas where the anthrax is endemic. We don't normally use, but there is a vaccine which is available against anthrax. Also, there's a vaccine available. Brucellosis, it has already recently been introduced in our UT. We use strain 19 vaccine. So ET is normally we use enterotoxemia as a regular vaccination, which is being used in our duty, and the tetanus vaccine, it can be used as and when the need arises, but otherwise there's ET and brucellosis is a part of our vaccination program which is being followed at UT level as well. So this is the first vaccine which is a clostridial vaccine, Clostridium perfringens. It's a bivalent type C and type D type of this, and there is another tetanus toxoid. These two vaccines are mixed together. We simultaneously use them. Then there are C and D types of preferences and tetanus. There are other vaccines, 7 and 8 way vaccines. This is a 3-way vaccine. There are other way vaccines also available, but these are not usually required. In mostly we found type C and type D type of disease we find in sheep and goats. So normally we recommend these C and D types of vaccines, Clostridium perfringens vaccine along with tetanus, or it can be without tetanus also. It depends upon a particular location. You can use it without only C and D type can be used. Then vaccination schedule here. This is the usually ET vaccine is the first vaccine which we need to give to a newborn, because newborn is also susceptible to the disease. Actually, there are different. There's a different schedule of this for if you or a doe is vaccinated during pregnancy or not. Normally, what we follow the vaccination schedule in case of the Clostridial or ET is that usually ewe or a doe, which when she's pregnant, we vaccinate that ewe or a doe 2 to 4 weeks prior to parturition. Prior to the date of expected parturition, that is vaccinated around 2 to 4 weeks prior to that date of vaccination, because what we want is that it should confer a passive immunity in the newborn. If the female which is vaccinated, if it gives birth to a newborn, and normally we must remember that we need to give booster doses. In case of the Clostridial ET vaccine, we have to give booster doses 3 to 4 weeks apart. So females giving birth to the first time should be vaccinated twice in late pregnancy, about 4 weeks apart. And if the lamb or a kid is born out of a vaccinated dam, if a dam is vaccinated and she gives birth to a lamb or a kid, that lamb or a kid can be vaccinated at 4 months of age. There's no need to vaccinate that lamb or a lamb kid immediately after birth if the mother of that lamb or kid is vaccinated. If this mother is not vaccinated during her pregnancy, the offsprings which are born from a non-vaccinated dam, they have to be vaccinated immediately within 1 to 3 weeks of birth. And then we have to give them the booster doses also. To lamb or the kid, we have to give them booster doses 3 to 4 weeks after the first vaccination. So that thing has to be kept in mind, that what is the schedule of that ET vaccination. It has to be followed like this. Number second is Capri pox vaccine. In case of Capri pox vaccine, we can use, there are available, both live vaccines are available and attenuated vaccines are available. And there are different strains which are being used for preparation. If we use live vaccines, they confer immunity for a longer period, up to 2 years, and inactivated, they have a short period of immunity. And normally with the vaccination schedule, what's being followed, we vaccinate usually the recommended age of vaccination of a newborn, when a kid is born or lamb is born. Normally we, after 4 months of age, we recommend that the vaccine should be given, because there are two reasons for that. One reason is that in case of the newborn, there can be maternal interference of antibodies, because there are antibodies which are being produced. I told you that which are passive immune responses, there that antibodies which are transferred from a mother to the offspring, that they're called maternal antibodies, and these maternal antibodies can they interfere in immune response. So that's one reason we don't give usually except these ET vaccine. We don't recommend a vaccine below 4 months of age. And the second one is that even though the immune system has started developed in the womb itself of a newborn animal, has started in the womb itself, but it takes so to proper development of immune response takes about 3 to 4 months, and at 4 months of age the immune system is properly developed, and if we'll vaccinate that particular lamb or a kid, it will give proper results. It will be immune response will be produced in that case, and the animal will be protected from a disease. So that's why we recommend it after 3 to 4 months of age or older. So once we talk of the Capri pox vaccine, regular vaccination has to be followed. if we are using, even though the protection is for 2 years, but we recommend, the recommendation is normally that in case of, if we are using live vaccine, live attenuated vaccine in our area, then the vaccination has to be carried out annually. But if we are using killed vaccine, then it has to be repeated twice in a year. Annually, the vaccine has to be given killed, since the immunity of this will be short duration. In case of the killed vaccine, the immunity will be of short duration. So it is recommended that the killed vaccine of capripox should be given biannually. And if we are using a live vaccine, then we recommend it as vaccination to be followed annually. So usually the vaccination, it is advisable that to vaccinate after kidding season or before the onset of breeding season. And animals in advanced pregnancy should not be vaccinated with live vaccines. So usually these are the queries usually which we get, that whether we need to we can vaccinate a particular animal during pregnancy or not. I think the answer to this is that if we are using a killed vaccine, that's a safe vaccine. We can go for that vaccine in pregnant animals as well. But if in case of live vaccine, there are chances of, and particularly of there may be some vaccines which, or some agents which can cause abortion. So we need to avoid those kind of vaccines, abortogenic, yeah, or live, particularly live vaccine which has a problem of reversion to that. And particularly in large animals, we avoid vaccination in the last month of pregnancy, because at last month of pregnancy, we need to restrain for the vaccination. We need to restrain the animal, and during the restrain the process of restrain, uh, at that time the animal may get injury, and it may affect the newborn, it may affect the fetus. So can be avoided in the last month of pregnancy. Otherwise, we can recommend the vaccination also using the kill vaccines in pregnant animals also. Then again, there is a PPR vaccine, which is usually a vaccine which we use in case of goats against PPR disease, which is disease of goats mainly, even though it can occur in sheep as well. But normally we recommend it as in our Jammu and Kashmir. We don't have in Kashmir Valley, we don't have the goat population to that level as compared to, we have sheep population, higher number of sheep are available here. But in some pockets of J&K, like in Poonch, Rajouri, there are some districts where the people rear goats as well. So during the time of summers, they migrate from their areas to the alpine and subalpine pastures. And that time of, even though in Kashmir Valley, even here we also vaccinate them against the PPR. So this is the important disease against the vaccination is being done here also. So we normally have live vaccines. They are mostly used against the PPR, which is cell culture derived vaccine, and it gives immunity for a period of 3 years. So this vaccine is recommended for again use at 4 months and above. Uh, it's advisable to vaccinate after lambing season or before onset of breeding season. Then vaccination of animal in advanced stage of pregnancy is not recommended. It's a live vaccine, so we don't recommend this. So this is a vaccine which is available against. We have a blue tongue vaccine. Normally, we do not use in our part. We do not use in UTL Jammu and Kashmir. We don't use blue tongue vaccine. As per our schedule, but it can be used in sheep and goat wherever the blue tongue disease is prevalent. It can be used, and we have live attenuated and killed vaccines which are available in case of blue tongue vaccine. Then attenuated vaccines are cheap and easy to produce, and but they have a risk of reversion. I have discussed about these things. Killed vaccines have shown the efficacy also and cannot be transmitted to other. I've discussed, but here the vaccination schedule for blue tongue is that again the lambs they should be vaccinated about 3 to 4 months of age and close to weaning time. Uh, and then herd should be vaccinated annually. Then we recommend, in case of this annual, this vaccination is recommended approximately 3 weeks prior to breeding or after lambing season. Then there is another disease, foot and mouth disease. Uh, Foot and mouth disease is not, it doesn't cause that much of clinical manifestation in case of sheep and goat. It causes mild lesions and fatal lesions, or in buccal cavity, then can be very few lesions. But as a part of the national eradication program of FMD, it's mandatory that we vaccinate the small animals as well, those small animals, they can serve as a source of transmission to the larger animals as well. So if we are going for the national eradication program, we have to take care of this thing also, that we vaccinate the small animals as well, because we don't want any kind of source to be there which can serve as the organism can survive, should survive in any possible reservoir. So that's why it's recommended that in sheep and goat also we use this FMD vaccination. Though it's mandatory because of that thing, control program, the foot and mouth control program. We use kill vaccines in case of this, even though subunit vaccines are available, but they generate a little amount of cell-mediated immunity, and keeping in view that thing, only kill vaccines are available in case of FMD. So again, here the primary vaccination is done at 4 months of age and above. Then the booster doses are repeated at 3 to 4 weeks apart. Then the regular vaccination is being done biannually. Here we recommend it at in the month of March and in the month of September. So it is to be done biannually. The vaccination has to be done. Then there's a rabies vaccination against rabies, but we recommend it's not routinely being done, but it's recommended that whenever there is a rabies infected area, or livestock has the access to wooded areas or areas frequently carriers of rabies, dogs are there which are the carriers of, there's a probability that they are or there's a this they are at risk, livestock is at risk, we feel the livestock is at risk. So at those areas where we can recommend this rabies vaccination. The cost of rabies vaccination has to be it's available here also in our UT. It's available in the government departments as well as subsidized rates. So we can use it as a prophylactic vaccines are also there, or it can used as a therapeutic vaccines are also there. Only inactivated vaccines are licensed for use in case of rabies. Then primary vaccination is done at 3 months of age and above. Booster doses, we need to repeat the booster doses since it's a kill vaccine at 3 weeks apart. Then the immunity is conferred. Here, immunity is conferred for 36 months. However, keeping in view the seriousness of the disease, it is recommended that animal vaccination to be followed in endemic areas. So, and in case of if an animal is bitten by a rabid dog, then we can have the proper schedule of the vaccination, which is there is a vaccination schedule which has to be followed there. Then there can be the vaccination there. Even though we go for the vaccination, it's not always there. We cannot guarantee that the animal will be protected against a particular disease, because the successful vaccination depends upon so many factors. There are n number of reasons which are responsible for vaccination failure. So keeping in view those n number of reasons, it's possible that even after vaccination, the animal may not be protected against a particular disease. But that does not mean that we will not vaccinate our animals. The chances are very rare, but we need to take care of, if we'll take care of these things, these reasons, if we know the reasons of a vaccination failure, and if we work on that during our vaccination program, we just try to avoid them and just try to figure out that what things we need to take care of, then I think that will lead us to a successful vaccination. One thing I forgot to tell you before this is that it is normally recommended that if we go for the vaccination in a particular herd, it's normally recommended that we go for the deworming first. We should first deworm a particular animal, and then only we should go for it's advisable. Why it's advisable? Because if a particular animals, they are close grazers, they graze they are at risk of developing the parasitic infestation, be that ectoparasitic infestation or endoparasitic infestation. And in that cases, I am sorry that I'm not able to, I'll be taking up the comments. There's in chat box there are so many comments, but I think I'll complete my talk, and then I'll just discuss.
Yes, sir, please. We will
Yes,
take up the questions at the end of your presentations.
Okay. I'll try to answer them to the best of my capacity.
Yeah.
Okay. So there are so many factors which affect vaccine efficacy. I'll discuss a few of them. Like there can be contamination. if we are purchasing a particular type of a vaccine from a trusted supplier, it won't be contaminated, but it has to be, we have to make sure that it's not contaminated. There's no sort of contamination should be there. If it is a trusted supplier, then there will be no contamination. It has to be from a trusted supplier. Then storage is important. Storage, by storage we mean that cold chain has to be maintained. I'll discuss about the cold chain after this. Cold chain means we need to store it at around 4 to 8 degrees Celsius. If we store it above that Celsius, there are chances that it may get the damage, the protein or this biological material which is within that vial, that may get, that will not retain its efficacy. So if the cold chain is not maintained, that can result in probable this vaccination failure. So if it is exposed to directly, it's exposed to heat or sunlight, so that can also result in this. And expiration, if it's expired, so naturally if a thing is expired, it's no longer is validated, it's no longer can be used. So it is better if we store that a particular vaccine according to the written indications. We must read the indications which are written on that particular vaccine, where to be stored and at what temperature. Those indications have to be followed in letter and spirit. So that thing has to be kept in mind to avoid this vaccination failure because of the storage problem. And it has to be used before the everything needs to be used before expiration date. So that applies to this vaccine as well. Then there's a procedure. There's a proper procedure that how we go for the vaccination. We have to use a proper dosage of vaccination. We cannot underdose it, because normally a proper dose is recommended. When we go for the vaccination trial, we use different doses at that particular, and we find through those trials or through in the research in the laboratory, the exact dose is the dose at which we expected a maximum amount of immune response. So that dose has to be maintained. If that dose is given lesser to that, a proper immune response will not be generated. If higher dose is given to that can generate an immune response, but at the same time that can overburden an animal, that can cause stress to the animal, because we are giving too many antigens to that animal. So that's also not recommended. So a proper dosage has to be given to the animal. It must be given. For example, if it is recommended 1 ml, we must ensure that 1 ml of that particular injection or that particular vaccine should go appropriately in that animal through a proper route. Whatever the route of administration is recommended, we must follow that, and that must, otherwise that will be of no use. Again, we have inappropriate sterilization. If the sterilization of, if we are using a particular needle which is not sterilized, it may cause infection to that animal. It may cause a kind of bacterial infection. It may cause sepsis and other things. So we have to follow proper sterilization protocols. Then so we should not miss particular animals. Once an animal is vaccinated, we can use some way of just indication that it has been vaccinated. It can be a tag or it can be anything else, but animals must not be missed. And the vaccine, if a vaccine is deposited in fat, that again will result in low immune response. So we must ensure that it is deposited, the vaccine is deposited where it is intended to be deposited. If it is intended intramuscularly, it has to be done that. If it is intended to be deposited in the subcutaneous, it is recommended it has to be like that. If it is intranasal, it has to be used through intranasal route, or if it is oral, then it has to be given through oral route. So those things are to be kept. So these things can be managed only and only when we have trained personnel or proper vaccination procedure is being followed. Only when we have trained personnel at the field level. Otherwise, we have so many new personnel at those who do not have, even who do not know ABCD of vaccination. They go and they carry these. Sometimes they carry these vaccines in their pockets, and they go and then they use a single kind of this syringe to vaccinate 10-20 animals. They use every kind of inappropriate procedure to vaccinate the animals. So that thing has to be avoided, and that you must remember that thing. These are some of the issues of why vaccination can fail, or what are this. The other thing is the stress factors. The animal must not be in the stress. Everything in our system, in our body, will function appropriately and to its optimum level when there is no stress. If there is any kind of stress in the body, stress can be of multifactorial. It can be a nutritional stress, it can be a stress due to a disease, it can be some other things. But there must not be immune that can lead. These all stress factors can lead to immune suppression. Immune suppression means the immune system of that particular animal is not working to this optimum level. It's working to below optimum level. That's known as immune suppression. So the immune suppression can be uh, because of so many reasons. Number one reason is nutrition. If a particular animal is not being given a proper nutrition, it will be in stress, nutritional stress, and there will be an immune suppression as well. So, if an animal is healthy, if an animal is not weak, if it's not emaciated, it should not be emaciated, it should not be weak, it should be in a proper health and nutrition. Then only we can, expect that better results of vaccination. The animal, in case of infectious agents, if there is already an infection which is there in an animal, he is already under the stress, so he is already developing an immune response against that particular natural infection. He will be under stress. So if we'll vaccinate that animal who is already having some kind of infection, so that will again not be a good thing. It will again lead to just it will not be a proper vaccination, which we call as. So we must ensure that a particular animal is free of infectious agent, is free of a particular disease causing agent. How we can ensure that? We cannot, in case of subclinical infections, we cannot ensure it, but clinically that animal should be healthy. We must ensure that clinically the animal should be healthy. So only we then we can vaccinate. And there is maternal antibody interference. So maternal, I told you that maternal antibody is after the newborn baby's kid is or lamb is born, then there can be maternal antibodies. If we vaccinate a particular animal against this maternal antibody can interfere. So we must ensure that there's a proper age for vaccination. That's what I told you. After above 4 months of age, you can vaccinate. So that thing has to be kept in mind. And there are certain other if there's a mycotoxin in the feed, that can again, there are some certain mycotoxins we can lead to immunosuppression. So it is to be ensured that they are fed with those kind of a feed which are free of mycotoxins, and there is no immune suppression in the animal. And these are the factors. There are other factors also, but I have tried to discuss only about important factors which are there for a successful vaccination, which has to be kept in mind. And I missed one point to discuss that thing in detail, is that uh, we must deworm a particular animal before vaccination. It's a proper. We must carry. Should use. Go for mass deworming before going for vaccination. Particularly, we can use a week, at least a week before vaccination. If we are going for a vaccination, just a week prior to that vaccination, we must go for antihelminthic dosing for that, so that the parasitic load, if it is there, it is reduced. And otherwise there will be again the proper immune response will not be produced there, and will be in stress, and will that already will be in stress because of that parasitic infestation. So it's advisable that if we go for proper deworming before vaccination. Hello, am I audible?
Yes, sir.
Yes, you are audible.
Then we have as I told you, that cold chain is to be maintained. Then we should not exceed. The vaccine should not be kept above 8 degrees Celsius. We recommend that it should be kept it should not be kept at this freezing temperature, but it should not also be kept at the same time above 8 degrees Celsius. Normally, we aim at 5 degrees Celsius. Excessive heat or cold can damage the vaccine. So ultimately, it will result in reduced potency. So it will not be that potent. So that thing has to be kept in mind. That's what we mean by maintenance of cold chain. Vaccine no longer it will not be licensed if the cold chain is not maintained. So cold chain depends upon three elements. that it will only be maintained if we use effectively trained personals. Dr. Ashok was telling me that we have a separate training institute for sheep breeders. I think we need to sensitize sheep breeders also. We need to train them as well. What are the proper vaccination protocols? They must also know about these things. And our own personal livestock assistants and other who are working at the field level, they must be made aware about all these, even though they seem to be very small things, but they are very important things on which is a successful vaccination program will depend on. So effective trained personnel is important. Then appropriate transportation and storage equipments. We need to have, normally what we do is that we ensure the proper quantity of vaccines are available at our centers, but we forget about the proper transportation mechanism which has to be put in place to transport these from one area to the another area. So we don't focus on that, but that's equally important as that keeping available the vaccines is, that we must have appropriate transportation, appropriate cold these refrigerated these vans, cold vans available which can maintain a temperature. We have to focus on that. And at the same time, once we are going to the field from a particular center, we are going to the field, we must ensure that these vaccines are being carried at a particular temperature. We use that igloo boxes we use that the vaccine has to be kept, and we has to use a proper thermometer has to be kept there where we can monitor the temperature. It has to be monitored twice a day. So all these things need to be taken care of. They have to be considered. Then efficient management. Only if we'll use the trained persons, we'll use appropriate transportation, storage equipment, then only our management can be efficient. And ultimately, it's only the efficient management which will make the vaccination program a successful one. Here, what we do is that we have to use a dedicated fridge, dedicated vaccine chamber for storing the vaccine. And it is to be ensured that we maintain a proper electricity supply. Otherwise, what happens usually we'll leave a fridge as such, there will be no electric supply for a day, but we'll assume that we have kept that in the fridge and they'll be okay. But we have to ensure this thing also, that there's a proper electric supply to that fridge also. And it should not be haphazard, it should not be full, it should be kept only up to 50% or 60% of it should occupy the space, fridge should be occupied. Then every vaccine places vaccines in the clear, they should be clearly labeled. The plastic mesh baskets must be there, they should be labeled by group vaccine types. We should regularly defrost and calibrate fridges regularly. Normally, we use fridges in our labs and other things which are decades-old fridges. We never calibrate them, but we must ensure, if we are keeping them in our at our own place in fridge, we must ensure that that fridge is maintaining a proper temperature. We must calibrate that fridge regularly and use defrost procedures whenever needed, particularly in winters. Then ensure backup facilities. So even sometimes what happens is that there is a fridge which will because of some reason or other it will fail. It will go out of order. Then we must ensure that we have a backup supply also there. So there must not be a period between when there is a breakup in the fridge and then we don't have a backup available there. We must ensure that we have a backup also available there. We must also ensure this thing, that no food or medical specimens should be kept along with that thing. If a fridge which is there, if it is dedicated for vaccine, it has to be dedicated for vaccine also. We must not use for this fridge for other purpose like food or the medical specimens. And we must not place the fridge in the direct sunlight or near the heat source. Vaccines should not be removed from original boxes until they are ready to use. And we must not, care has to be taken that we must not ensure sometimes knowingly or unknowingly we place somebody might place them in the upper most region which we call as the freezer of the refrigerator. We must avoid keeping them in the freezer. They will lose the potency once they are kept in the freezer, where the temperature can be around -10 also. So they have to be, care has to be taken. Though we must use these maximum and minimum thermometers. we probe, it must be placed in the center of the fridge. We must record temperature at least, if possible, twice a day. Reset has to be done daily. Calibration is recommended. And if there is temperature, if it's outside recommended range, then necessary action must be taken. Then there are some vaccine related adverse reactions also. Usually the vaccine minor reactions due to vaccines, which are not to be reported. There are some, if we use a vaccine, there can be a mild reaction to this, but it is treatable at home. It can be managed at home. It should not be repeated. For example, there is a local reaction, pain can be there, swelling can be there, or redness can be there, the local inflammation can be there. So the treatment for this is that we can use a cold cloth at injection site, or we can give a paracetamol tablet. That's what we do when we vaccinate our young ones or kids at home. We give them a dose of paracetamol, and next day they'll be doing fine. So that's what's recommended. That's a local thing. Another mild reaction in case of the vaccines can be fever. It can go up to 1 or 2 degrees increase in fever can be there. Then at that time we can give extra filbert to them. We can give spongin, can be given. Even paracetamol is recommended at that time. Or there can be irritability or malaise and other. At that time we need to manage it at that locally, and there's no need to panic at that particular time. It's not a thing which is to be reported to higher authorities. We can manage it at a local level. These are the normal reactions which can be these are normal physiological reactions which can take place once the vaccination is given. I discussed about the minor reactions which are there, which need not to be reported. There are other events, but which are very rare in case of the vaccines we are using. There can be some events also which need to be reported immediately and which need to be investigated. You can report immediately to your higher ups. It depends who the higher up is, but the proper procedure has to be followed there. You cannot report it. There is always, once you are working in a particular system, there is a proper way of reporting. The proper way of reporting, their thumb rule is that you must report it immediately to your higher up. He will then report to his higher up. Then this is the chain which we must follow. So the events which have to be reported, even though they are very rare, but they can be occasionally at very rare events can be there, that it can be a death, that they can cause death or disability or a serious or unusual event other than what I've discussed in the mild reactions. Then if the events are occurring clusters, if there are, and if a particular animal is having that event, we can manage that. If these events are occurring in clusters in bulk, then we have to report that. If there's anaphylaxis, there's hypersensitivity and anaphylactic reaction, if it is there, it has to be reported. Toxic shock syndrome, then acute hypersensitivity reaction, encephalopathy, sepsis, any event where vaccine quality is suspected, or there can be acute facial paralysis. In nutshell, the events other than the mild, which I discussed about the mild, if you notice some untoward or a particular thing which can be a serious thing, then only you need to report that. But these things are very rare in case of vaccines. These are very rare. These are not usually reported. So this is again, this I have discussed this in a tabulated form. Some vaccines, and along with their route of administration and along with their schedule, I've discussed this. This is briefly the vaccine transport. If you are carrying it from your own center to the field level, then you can carry it in your igloo box. You can use ice packs also. Normally in this we use ice packs, and this is the proper way how we can keep the vaccine. We have to keep the frozen ice packs at the bottom. Then there will be there will be vaccines, and in the middle of the vaccines we have to keep our temperature gauge, which is a minimum and a maximum this data or a data logger you can keep there, and on the top also you have to keep the ice packs. So this is how you must carry, and you have to ensure that you just monitor the temperature every 2 hours, 2 to 3 hours. You monitor the temperature. Whether sometimes what happens that we carry it over a long distance, the ice packs, they have usually melt down and they are no longer able to keep a proper temperature, which is recommended there. So we have to ensure that this proper temperature, and we have to monitor this temperature regularly. Then expiration date, we have to, if it is written that it has to be used before expiry date is August 16, so we should not use on 17th of August. That's what is recommended. So likewise, if it is 31st of August, we should not use that on 1st of September. So that has to be kept. So this is how sometimes you get a lyophilized powder, then you have to dilute, then you reconstitute that. But dilutes, they are not interchangeable unless it is specified the manufacturer. We must, we must focus what has the manufacturer recommended. We must, follow that. It must be strictly as per the manufacturer's recommendations, because the manufacturer is the one who has prepared it, and who knows that what kind of a diluent he used and what kind of a other lyophilized powder he's used. So whatever he has recommended, we must follow the manufacturer's protocols strictly. There are multi-dose vials recurring reconstitution. We must, once we have a particular kind of a vial which is a multi-dose vial which has multiple doses in it. It can be so we have to mark each multi-dose vial with date. When we open it first, we have to mark that. Sometimes what happens that we may not be able to use that whole of the vaccine vial in a single day or at a single particular moment. So we have to mark each multi-dose vial with a date. First open, some vaccines expire within a certain time after opening. So it is written there it will be that sometimes the vials are there. It is written there. It must be used after opening. It must be used within a day or so. So it has to be kept in mind that what the recommendations are there. Accordingly, we must follow. So we must ensure that the date of opening is written, and it must be utilized within the recommended time frame. So this may not correspond to the expiration printed on a vial by the manufacturer. These are two separate issues. One is the manufacturing expiry date, and the another thing is that in case of the multi-dose vials, we have after what period of time after opening it we must utilize it fully. So we must ensure that thing also. It varies from manufacturer to manufacturer. So this is certain key points which we must keep in mind, that we must assign responsibility of handling vaccines to trained staff. We should have a backup staff, primary trained staff, and a backup staff also. We must regularly check vaccine shipments, store vaccines in appropriate type of refrigerator, appropriate place in the refrigerator, which I already discussed. Check up expiration dates and rotate stocks. Check temperatures. We should document temperature. Take action if the temperature is out of range, and have routine and emergency vaccine handling plans. Then if any kind of this untoward event is there, which you find out that it is to be reported the higher, then you should immediately, if it is of such serious nature, then you should immediately inform your higher ups of the incident, treat that as a serious untoward incident, or suspend all the vaccination until resolved. If in case you find that some event which is of some serious nature is arising out of the vaccination, you must ensure that at that very point you must suspend all the vaccinations until the event is resolved, and immediately either inform your higher ups so that the necessary steps to resolve this can be taken up. So there are some questions, few of the queries which people usually ask, that what is the ideal age of vaccination. I have discussed that you must vaccinate around 4 months of age and above is the ideal age of vaccination and in vaccinating an animal, except in case of the ET vaccine, which you can give it, below that age also. Booster doses it's not required for every vaccine, but it is required usually in case of the kill vaccines, where the immunity level, immune response is not that much. In those, particularly in case of kill vaccines, there's a recommendation that booster doses must be given. But in case of live attenuated vaccines, booster doses are not usually required. So the other thing is that how we can identify a particular successful vaccination. Successful vaccination, to identify a particular successful vaccination, you need to have a laboratory backup. Since I am a person from a research background, So I will recommend that we cannot, we cannot just say that a particular vaccination has been successful only on the basis of the fact that the animals they have not they do not get the disease as such, because we never know whether they are at the risk or they have received they have got an exposure with the infectious agent or not after that vaccination. We never know. But what one thing is that if at the laboratory level, if we want to know the successful vaccination, there is only one method, which is that we antibody titer, which we can just check of that animals. If a vaccine herd is vaccinated, we usually do is that we take the serum sample at 0 day, that means prior to vaccination, and we check out if there is some remnant antibody titer already, their animal can be infected of some natural infection, there can be already an antibody titer. So we must ensure that at that point of time, we must ensure that also. Then after, say, after vaccinating an animal, after 2 to 3 months, we can again go for the sera collection and we can check the antibody titer. If there is a proper antibody titer against that particular disease or infectious agent, only then we can say that the vaccination has been it has produced its effect. Otherwise, in some animals, I discuss about if the animal is weak or immunocompromised or other things are there, then there cannot be proper immune. Or if there is a problem with the vaccine, if there's a manufacturing if it is contaminated, if there's a manufacturing defect or other things are also there. So again, then it will not be able to produce the immune response which it is capable of. So only thing is that the immune response, proper immune response, which is the antibody levels, proper titer, antibody titer, which we can find after 2 to 3 months, protective antibody titer, which we need to find out, which will give an indication that it has been a successful vaccination. Other than that, there are clinical signs. If an animal is, we can, just, it's a common sense that if the animal is not getting a disease after vaccination for a year or more, we say that it's already vaccinated and there is no there's no outbreak. So that we, but to be sure about a successful vaccination, only by means of this laboratory testing we can be 100% sure about a successful vaccination. Deworming mandatory? Deworming, I think it's advisable to deworm your animal before going for vaccination, at least 1 week before you go for the actual vaccination. It is recommended. And what's the first vaccine? I think ET and enterotoxemia vaccine is the first vaccine. Usually, after the birth, 1 to 3 weeks of age, we recommend enterotoxemia vaccination to be given if the ewe or the doe, pregnant animal, is not vaccinated, and that gives birth to a lamb or a kid. Having said that, this vaccination itself, it's a I know big topic. It's a vast topic, and vaccination. I'm talking from this part of the country, and maybe in other states, there may be some other diseases also against which the vaccines are available, or this. But I'm not sure, pretty sure about that, to be honest. What I'm sure about is that what vaccines we use here, or what are the types of vaccines we use in our UT against common infectious agents. Yes, I have now discussed about a few other 1 or 2 vaccines. One is the strain this against the brucellosis. Recently, in our UT, the vaccination against the brucellosis has been introduced, and it is we use strain 19 vaccine against brucellosis, and we generally we vaccinate the female animals above 6 months of age. We vaccinate. We don't vaccinate in case of the brucellosis. We don't vaccinate the male animals, but female animals we vaccinate, because we have a good prevalence of brucellosis in Kashmir, Jammu and Kashmir. So that's why the vaccination against brucella has been introduced, and keeping in view the zoonotic nature of the disease as well. It's very imperative that we start the vaccination. So it has recently been started in our area, and there is anthrax vaccine, which is spore-based vaccine. We do not usually go for the. It's recommended only in endemic areas where we suspect where there's a disease. But here, In our UT, we don't go for anthrax vaccination as such, but it is a vaccine which is available and it can be also used if at places where the disease is endemic. So it can also be used. So these are other two types of vaccines which are also there, which can be used in animals.